going to be confused. Oh, there we go, being live streamed.
Meeting is being live streamed. Awesome.
All right.
Okay.
Great. Okay, so I'm gonna take that again from the top, I think is what we're gonna do. Okay, hello everybody and welcome to Elasmo Week 2021 live Q&A. I'm your host, Vicki Stein, a science journalist and science writer. I'm thrilled to be here with a group of incredible Elasmobranch researchers and experts, many of whom were presenters in this week's talks. So remember, if you missed any of the talks or if your interest is sparked by the Q&A, you can find all of these speakers and their wonderful events recorded on the Elasmo Week YouTube page. So yeah, I think we're going to dive right into these shark-rich waters and have our panelists for the event introduce themselves with their name, their area of expertise, and their favorite Elasmobranch. Yes, that's right. We're just jumping straight into the drama. Give me your best spicy best shark takes, or ray or guitarfish or whatever takes. So I'm gonna just start with Dr. Schiffman.
Hi, everybody. My name is David Schiffman, coming to you from Washington, D.C. The past Elasmo Week folks might recognize me as the host from last year. This year I was a presenter, and my expertise is interdisciplinary shark conservation. And favorite shark, gotta go with #BestShark, the sandbar shark. Follow #BestShark on Twitter and Instagram for years of me talking about these and years of my colleagues telling me I have picked a boring animal. There's also a #YawnShark, that's a reply.
All right, who wants to go next?
I can go.
Okay.
Yeah. My name is Lisa Whitenack. I'm at Allegheny College, which is about an hour and a half north of Pittsburgh, Pennsylvania. So nowhere near an ocean. I am a shark paleobiologist, which means I study both living and extinct sharks. Megalodon's super dead. I'm just going to put that right out there. And my favorite living shark is the frilled shark, mostly because they look like Muppets and their teeth are little tridents that are curved backwards like a snake fang. And I've got my hands stuck in their mouths, dead ones, but multiple times.
Great. All right, Maria Rita.
Okay, so if I can go, so I'm Rita and I'm from Portugal, which means that now is 11 p.m. here, and it's pretty late. And I have been working with regarding the impacts of climate change in sharks. And my favorite shark is the whale shark. So yeah, so you can go. Yeah.
All right.
Hello, everybody. My name is João Paulo. You can call me John if it's easier. I don't know. João is difficult to say, I know. I work with systematics of chondrichthyes. My work involves the competitive anatomy of the paired fins within this group. So I'm trying to understand the evolution of the paired fins. And my favorite shark, it is a really hard question, but I do like the tiger shark. I think it is beautiful.
Awesome. All right, great. So I think, we've got kind of a bunch of really quick questions this week. Some of them are like definitions of terms. Some of them are little snapshots of particular animals. So I was hoping we could just kind of run through those in sort of a lightning round kind of a thing. If you've got an answer for a question, please just jump in. And if we get multiple people talking, I'll try and choose someone to start, but you're all welcome to add on to each other's answers. So the first one is, what are chimeras? Anybody want to take that?
Yeah, okay, I can take it. So when we look at chondrichthyans, which is the big fancy science word for sharks, skates, rays, and these chimeras and ratfish, there's fishes whose skeletons are made out of cartilage, so similar to the stuff at the bridge of your nose. Scientists like to sort things a lot, and so we sort them into two groups. We have the sharks and the skates and the rays, so the elasmobranchs, which Elasmo Week is named for. Then we have this whole other group of fish that are called holocephalans, and that includes your chimeras, your ratfish. Water bunnies is one of the names for hydrolagus, one of the species in that group. You may also see them called ghost sharks a lot, so not actual sharks, but because they're made out of cartilage, right? We'd like to throw shark on the name. They're really cool. They don't have the gill slits that sharks have. They have a bone, or not a bony, but a covering called an operculum that covers that. Their teeth are really different. So tooth plates that are flat instead of lots of kind of sharp and multiple teeth that sharks change out from time to time. The chimeras don't do that so much. But yeah.
Cool. And where do we find these animals living usually?
There's a lot of them that are deep in the water, and I know I've seen some images online of folks catching them off of Washington in the Pacific Ocean as well. But I'm no chimera expert. That's where my
Cool.
knowledge stops.
Right. Awesome. All right. So the next question we got actually is, what are ratfish, which I think are relatives, right? Or maybe the same animal but different name.
Different, yeah, they're in the same group with that umbrella of holocephalans. And I think there are some, right, if you talk to someone who specializes in them, I think they tell you there's a, this is a chimera, this is a ratfish. In my brain, they're pretty darn similar.
Awesome. I love that there are experts in very specific things like that. That's the greatest. Okay, so the next question is, what is bioluminescence?
I think that it is a special property that some sharks have, for instance, and they can produce some light just to disguise themselves, so they are not seen by other predators, or else they can use that same kind of bioluminescence to try to prey other fishes. So it is a kind of way to disguise themselves, and it's really cool. So when I started studying sharks, for instance, I had no clue that they had that kind of thing. And when I got to know that, I was wondered. It is a really cool thing.
Yeah, I love the idea of like sharks with disco lights, or like sharks that are using light to camouflage themselves. That's very cool. Okay, is the pocket shark the smallest shark? That was a question that we
So
got.
I just had to look into this stuff. I write an Ask a Marine Biologist column for Sport Diver magazine and just did one about shark sizes. There are a bunch of sharks that are all pretty much the same size, that are pretty small. The pocket shark is right in that range. The dwarf lantern shark, or pygmy lantern shark, is commonly cited as the smallest. We're talking a few inches long, but there are lots of sharks that are all pretty dang small. And the pocket shark is well in that range. I do love pocket sharks. I got to meet the pocket shark holotype at a past conference. That means the individual that was first caught and described by scientists, it was in a jar of formaldehyde that was brought to the conference and passed around for people to take selfies with. So you can tell we're the cool kids at the party. But there are lots of sharks that are under a foot long, and that is one of them.
So we're talking like, if you had to show me with your hands, we're talking like a little teeny boy.
Yeah.
Oh, little.
Yeah.
Oh, that's so cute. Oh, I love that. And where do they live? Most of these teeny tiny sharks.
Typically, we're talking deeper animals. The animals that live in the deep sea are often smaller. There's more pressure there. There's relatively less food there, and being small is an adaptation to that. But there are certainly some larger animals that live down there as well.
Cool. All right. So this next question, I'm going to try and read out loud all in one go, but we'll see how it goes. Are the birdbeak dogfish shark and the spiny dogfish shark related? That one's real specific.
I think I'll need some help with the dogbeak shark. I cannot relate to the species. Considering that it is a popular name, but anyway, it Dynia? No, yeah. So yeah, I would say that they are related, and they are in the same order that we call squaliform sharks.
Mm-hmm.
Yeah, I just checked. Same order, different families. I had no idea. Had to look it up.
Yep, I totally Googled that while you were talking.
Nice. Love Google.
One cool thing about them is that they have some of the species within the order. They have spines preceding the dorsal things, and they can be used as a protection for the shark. And what else is cool about this group of sharks? They have a really cool process on the upper jaw that helps. How can I say that? That is exclusive to this group of sharks, and we can recognize them based on that kind of morphology of the upper jaw. So it's really cool.
Cool. All right. Are cookie cutter sharks good swimmers? I mean, I would assume most things that live in the ocean are at least a better swimmer than me. So there's that.
I don't know that anyone's like thrown one in a tank and tested it. I mean, if
I want to do that now.
I know. I was just thinking
Put
about
one in
like,
one of those like tubes for,
like I
you
have
know.
a flume. I could do this.
Oh yeah.
Or Brooke
that's the word.
Flaming could. I mean, they've got to be, right? So if they're chasing down, it's not like they're just happening upon a dolphin.
Right. So a
to
cookie
take
cutter
chunk
shark
out.
is a type of shark that will swim up next to something bigger than it and take a chunk out of it, right? So
Mhm.
presumably they have to be able to at least match the resting speed of one of those things.
Yeah. So there you go. Task for
Cool.
future scientist.
Yeah. Great. Okay. Do guitarfish flap like stingrays or swim like sharks?
These are some very specific questions here.
I know. Yeah.
Yeah, I'm having trouble also answering these questions.
The only guitarfish I've ever seen, it swam by using its caudal fin, its tail, like as a shark swims.
Mm-hmm.
There are lots of guitarfish, and I have not observed or seen video of the others, but they seem to have a more substantial tail than a typical stingray would, and probably I would guess that it's used for locomotion, but I am not sure at all.
Cool. I think it's important that we say when we don't know. Go
I
ahead.
I do think they use they caudal fin to move across the water. So other orders of rays, just like the rajiforms or medial body forms, their pectoral fins, they are much more developed. So they use their fins to move, unlike other groups of rays. So rhinobariforms, so the systematics of Elasmobranchs is really confused right now. So we used to have a group that we would call rhinobariforms, pristiforms, but now we have rhinopristiforms. So all these group of rays, all these individuals, they use their exoskeleton, their caudal fin to move across the water. That's it.
Cool. Okay. The last one for the lightning round here would be, what kind of teeth do manta rays have?
Very tiny ones.
Okay. Well, I think we're gonna talk more about filter feeding a bit later on, so we might maybe just leave it there. So actually, the next question that maybe we'll go into a little bit more is, what can teeth tell us about shark body size?
Yeah, so I study teeth. That's like my jam. So
Awesome.
yeah, it's depends on the shark, right? You have the biggest shark in the sea, which is a whale shark, and they have itty bitty little teeth because they're filter feeders. They're not using those teeth, and there's some folks that are trying to figure out exactly how useful those teeth are. So if we were trying to use tooth size as a direct, like, oh, this big, I have a big tooth, so it's a big shark, small tooth, small shark, that doesn't work at all. Even things like nurse sharks, which can be pretty big, I mean, not as big as whale shark, and they use their teeth for grasping stuff. Their teeth are still pretty small. What we can do with those teeth, though, to figure out how big sharks are, is if we already know, the relationship of tooth size to body size, we can do some math and statistical modeling to say, all right, we have these set of sharks that we measured their teeth on, and we know what this mathematical relationship looks like. Now I have this tooth that I found over here. I can plug that into that formula, figure out how big it is. Or for things like megalodon, right? We base it on something that we have alive now with the white sharks that are relatives, and go, well, this is the best approximation. You're muted,
Oh,
Vicky.
sorry, I'm muted. There I am. Okay. Can freshwater stingrays go into the ocean? And why or why not? I think we're talking about, what makes a fish a freshwater fish versus a saltwater fish. And, why can some seem to go in between and some can't?
They cannot. In fact, they don't have all the physiological specializations to go or to invade the marine water. And in fact, the freshwater stingrays, they are the only group of elasmobranchs that their evolution is closely related to the freshwater environment. We have other examples of or groups of rays that can invade the freshwater environment, but they are not specialized for this kind of environment. So it is really cool. But if you put a freshwater stingray in a marine environment, it will die eventually. Yeah.
Cool. All right, great. And what is the stingray shuffle and why do we do it?
I can do this one. Stingrays are called that because they have that stinger on their tail that if you step on their back, they don't miss. The stinger, the tail moves up really, really fast and stings, and the stingers are quite nasty, often serrated, often very sharp, sometimes have venom. But they can't swim up to you, next to you, and sting you. It's a reflex that's triggered by pressure on the back. So the stingray shuffle is if you're walking in like ankle or knee deep water at the beach, not picking up your feet like you would when you're walking normally, but dragging your feet through the sand. And that way you're scaring away stingrays. And it helps to, it makes it so you're not going to get stung in the leg and ruin your beach vacation.
Awesome. Okay, what are, I'm not sure I'm going to say this word right, teleosts, and what are they important for?
Who is asking these questions?
I was given a list of questions.
I think I can answer this one. Teleosts are, as Lisa was saying, we categorize animals, and so we have the cartilaginous fish, which are the fish that are cartilaginous, like the sharks and the rays and the chimeras. And then we have the teleosts, which are bony fish, which have, instead of cartilage, they have bones. So that's
great. That's perfect. I think these are questions from people who probably watched previous talks this week, and some of them are definitely like, I don't recognize that word. Please help, which is great. I'm excited about that too.
We're glad to see people continuing their learning.
Okay, so some of these get a little bit technical as well. I think people wanted us to talk a little bit about lateral lines and the ways that sharks detect things. So one person was asking, if the lateral line is used for water pressure, how do sharks detect electric fields? So is that, is there an additional sensory organ for electric fields or magnetism or electrical signals? And how do you spot that on something like a shark or a stingray specifically?
Yeah, sharks have, and also rays, they have what are called ampullae of Lorenzini, which are these organs that sense the electrical field. So they can sense they can feel electroreception with these organs. I don't know if any of you guys want to add anything to that.
So is the lateral line a different thing?
Yes, different thing.
Great.
Yes. So if you look kind of at this part of the shark, underneath the nose, you'll see these really big pores, and those are electrosensory pores and some other lateral line type pores, but a lot of them are the electrosensory pores, especially in a hammerhead where you'd see it all the way across.
Cool. All right. somebody was asking, what are some other cool extinct sharks? Which I think is neat. There's a sort of a related question. People always talk about the Meg being a really big extinct shark. Was there ever a ray of similar size? So extinct sharks, extinct rays, like tell me what's good.
Yeah, so with extinct, I'll do the ray one first because that's actually on the easier side. We don't know. And this is again because the fossil record is mostly teeth, right? That cartilage that we talked about does not fossilize very well, even though some cartilage in the sharks has some extra mineral on it to make it extra durable. But when we're talking about millions of years, right, and you've got more rocks piled on top of that, glaciers wiping things away, other tectonic type stuff, it's really detrimental or difficult for those fossils to stay fossils and not just get powdered and disappear. The stuff that's harder tends to last, like teeth. And so when we couple that with the the problem where the tooth size doesn't always match the size of the animal, it's really, I don't think we know if there were huge rays bigger than, say, mantas or something like that, or some of these other bigger rays that we see. We do have ray teeth in the fossil record and stingray spines, but again, it's hard to correlate those to size. Cool. Oh, I could tell you about so many cool fossil sharks. This is where visuals are good. My talk and then Jada and Annabelle's talk both show some really, really wonderful fossil sharks, including Helicoprion, which is not a shark, but actually is related to those ratfish and those chimeras that we should talk about, even though they look kind of sharky, we think. They're the ones with the buzz tooth shark, right? The one with the tooth whorl. That's the only part of the shark we have. So everything else is just conjecture and supposed, in terms of figuring out what they look like. And then in my talk, I showed one called Damocles, which was only about yay big, but it has this kind of, looks like a sword hanging over its head, thus the name Damocles. And it's 320 million years old or so. And it looks just like a shark with a big thing on its head. And they're so little and adorable. But yeah, the further back in time you go, the weirder fossil sharks get. Like that pre-dinosaur time is the best time.
Yeah, awesome. Okay. All right, so this one's kind of funny. Elasmo Week got a question on Twitter about an Octonauts episode that the question actor regards as sus because the whale shark's teeth in that episode seemed way too big and there was some kind of like spongy membrane at the back of the shark's throat. What do we know about a whale shark's kind of feeding situation? The question asker's words are, what is the general mouthy anatomy and function of a filter feeding whale shark?
I could take this one again, I guess.
Yeah, go ahead.
So yeah, the teeth on that, I love the Octonauts. I'm going to say that. I also have 2 kids, so we love the Octonauts in our house. The teeth are really small, itty bitty. You need a microscope to see their shape, so they're really small. So in that episode of the Octonauts, the teeth were definitely way too big, right? So that was more of just, this is a show geared towards preschoolers, but adults love it too, right? I always watch the Vegetables Christmas episode every year because it's hilarious. But yeah, the teeth are too big, absolutely. Those spongy things that they showed in there are supposed to be the filter pads. So what those do is they help kind of direct the flow in the mouth and kind of help separate out all of the krill and plankton and all that stuff that the shark wants to eat. So that part's actually correct. And I'm going to give a shout out to Laura Ferry, who is a shark biologist who actually is one of the scientific consultants for the Octonauts. And so she helped with a lot of that kind of stuff.
I love it. I love it. I assume there was some amount of they put the teeth in there so that you could see them, so that you're looking at a shark.
Well, so that they're smiling and everything.
Oh,
right?
sure. Yeah. I haven't really watched a lot of Octonauts, but now I kind of feel like I should.
You need to. It's great.
Yeah. Okay. Awesome. Maybe I'll recruit a younger cousin or something to hang out with me, but maybe
I watch it by
myself.
That's what I do.
That's awesome. Okay. Let's see. So what does interdisciplinary mean? I think that word came up a couple of times in some of the talks, and what's that about?
I can start with this. So basically, in the sciences, there are the natural sciences, which are physics and chemistry and biology and ecology. There's also the social sciences, studying humans, sociology, psychology, economics, things like that. And interdisciplinary means using more than one type of science. The sorts of global scale challenges that ocean conservation faces often requires using more than one tool at a time. Often this is big teams all working together. Increasingly, it's also scientists trained in more than one scientific discipline. How I was sold on this, I did my master's in pure marine biology and then did an interdisciplinary PhD, was if the problem is humans are killing too many sharks, the solution is not passing a law that changes what sharks do. It's passing a law that changes what the humans do, which means we need to understand the human side. Why do people do what they do? What do they know? What do they understand? What can be used to persuade them? What would not work? And that's often the sort of work that I do, and as part of why I'm in the DC area, is there's a lot of where science meets policy type decisions are happening.
Oh, I'm muted again. There we go. I have a question, I think, Dr. Schiffman, that's specifically, I think, about your talk. But it is if fishermen catch more sharks than big boats, why haven't rules changed?
Yeah. So the specific statistic for anyone who hasn't seen the talk yet is in the United States, since 2013, every year, more large sharks are caught and killed by recreational anglers, just regular people with rods and reels, than by our commercial fishing fleet, which just blows people's minds when they hear that. That the large shark there isn't important because most of the commercial fishing fleet targets smaller sharks. But why is that changing? Because no one knows it's happening. So much recreational fishing policy is based on the idea that who cares? It's not that big a deal. It's just a few people with a rod and reel, but it's tens of millions of people with a rod and reel catching tens of billions of fish a year. And that's only counting the fish that are reported, and that's only counting the fish that are intentionally caught and killed. As I also mentioned in the talk, lots of times fishermen release the shark, but after they handle it so roughly, that's a dead shark. But in the fisherman's mind, they've released it and they have not killed it. But it's then when the dead hammerhead washes up 45 minutes later at the beach, oh, that's not mine. I released mine. That's just a total coincidence that one the exact same size washed up. So why is little changing about this? It's just increasing. It's increasingly recognized that recreational fisheries can pose a conservation challenge. I believe the title of the talk I gave was an emerging conservation issue or something, which means we're starting to think of this. We're starting to take it more seriously. We're starting to do more research. We're starting to change the laws. But for a really long time, including when I started doing this, a lot of people thought this is really not a big deal. Why are you wasting your time? And it turns out it kind of is a big deal and it's worth paying attention to.
Great, thank you so much for that answer. All right, so this one is going to be another kind of a group question. Somebody was asking about, like, do shark researchers make a lot of their own technology that they use to study sharks? And I know there is some incredible tech out there being used to study sharks. I actually wrote an article a couple of years ago about the researcher called it a shark Fitbit. But you get a shark to swallow something that can track their movements and can track how deep they are and can track all kinds of interesting, like, temperature and all sorts of cool stuff. But there's just, the ocean is not a place where humans can just follow a shark around. They live underwater. It's very difficult to study any elasmobranch, really, compared to, like, I can watch house sparrows in my backyard all day. So what kind of technology have you used and have you created or have you loved that has to do with elasmobranch research?
I haven't, but for example, a colleague in my research lab, she built, I don't know the name of it, but it was, I think it's far. I don't want to say the name. I really don't know, but it's kind of like a cage where she basically annuls the magnetic field of the Earth, and so she built that. So I think that's kind of cool. I never did something like that, but she did, so I think it's cool to to tell you about it.
That's awesome. I think that's called a Faraday cage, right? To kind of nullify.
Yeah, I did. Yeah, that's it. Yeah.
Awesome. That sounds really cool.
So I'm more of a lab rat and museum rat than being out in the field. I do a little bit of I in the past have done a little bit of that, and the biomechanics work. So this physics and engineering stuff applied to how animals move and use their teeth and all that. In my PhD lab, we built lots of stuff. So we built, we used a catheter that was used to measure blood pressure in arteries and ran that through some PVC to measure suction pressure in nurse sharks when they were feeding. We built bite force meters. In my lab, I do, because I'm at a small institution with mostly with undergraduates, I do a lot of what I call kind of cheap science and a lot, or MacGyver science or Home Depot science, where I'm going to my local home improvement store and wandering around and like, all right, how can I build this thing? Because I don't have this $1 million machine that I got to use during my PhD. Can I build that out of PVC? And so I do the thing that my students are using to test puncture forces in shark teeth, PVC teeter totter that I've built with just some, 1x2s and these PVC pieces, and then a fishing scale, like you would hang a fish from, to pull up and measure force as that tooth goes down, which was something that my PhD advisor had gotten the idea for. So when one of my classes where we learn research methods and how to do this, I take them on a field trip to Home Depot. I go sit in a comfy chair in the patio section and say, all right, go figure out how you're going to do this and come back with your ideas. Best field trip ever.
I love that. I love the creativity.
I haven't just like built anything to study my fishes. I use some old techniques. So I manually dissect some sharks and rays to study their skeleton. And there is a really beautiful technique that we call clear and stain. So what we do, we stain the skeleton with a stain that we call Alcium blue. And after that, we just digest the tissues of the fish. So we use a kind of an enzyme that we call trypsin, and the fish becomes clear, and it is a really, really beautiful kind of preparation. Besides that, there is a new kind of a new method that we are employing right now that we call CT scan. So we don't have to manually dissect anymore a shark or ray. We just put the animal inside that kind of machine, and we get a whole 3D reconstruction of the fish. It is amazing, really amazing.
That's so cool. What's the advantage of doing that versus a dissection? Does it mean you get to keep a specimen intact, or does it mean you can do it without, is it like non-lethal, or it's still kind of?
Yes, because when you manually dissect a shark or ray, you can no longer use it for a taxonomic study afterwards. So there are some kinds of fish that are deposited in collections around the world that we call types. So they were the first individual collected for that group of organisms. And if you want to study skeleton of a type, for example, you cannot dissect it. So if you can use a CT scan, you can learn a lot about the anatomy of a fish or any other vertebrate, and without just like destroying it.
I imagine some of those animals are probably pretty hard to come by as well. It's like unlikely that you'd find a lot of them to study, so you don't want to ruin one specimen. I love the cat. What's the cat's name? I think you're muted.
His name is Chino. because he has the color of a cappuccino, so we call him Chino.
That's awesome. I know somebody is going to ask that question on YouTube. So, just answering ahead of time. It's a journalistic, standby. So you always ask for the name of the dog. You always ask for the name of the cat. Important. All right. Dr. Schiffman, did you want to add anything to the tech discussion?
I don't use as much of it, but another advantage of the CT scanning being described here is then those files you can copy a digital file, and then everyone anywhere can be looking at it. And you can also 3D print from that and make physical copies that you can hand out anywhere. So I've been taking advantage of some of my colleagues who do this 3D or the CT scanning, so I can make cool models on my 3D printer at home.
That's awesome. I want a 3D printed megalodon tooth or something. That sounds great. Okay, so this is another kind of a researchy sort of a question. How do we know the temperature changes in the water globally? And how do we know that the acidity of the water is changing globally? How do we measure those kinds of things? That may not be a question in the wheelhouse.
There's an oceanographic sampling gear is the short answer. There are buoys with sensors that are just put everywhere. Then research vessels take samples. At the University of Miami, where I was, we were experimenting with attaching those samples to the cruise ships that cruise around the Caribbean. So there are physical devices that measure that.
Awesome.
You can also use the rock and sediment record if you want to go back in time, right, other than the previous. So there's lots of chemical markers, and you only get certain types of minerals under certain conditions. So all of that kind of can help us figure out what happened before we started collecting this data in modern oceans. And so if we want a long-term kind of look at it, we can look at all the geologic stuff.
Right. So if I understand correctly, oftentimes in the ocean, layers of rock build up, over time. So you can look at a sequence of things if you dig down deep. Is that right?
You can also use the ice cores, and you can get the bubbles, the air bubbles that were trapped there, and you can also measure the CO2, for example.
Cool. So similarly, layers of ice building up year by year, time, over time. Super awesome. Okay. This is a question that came in from YouTube that I just think is really interesting. With anthropogenic pressures, what would you say is a likely pathway for sharks to evolve in the Anthropocene if they could keep up with the degree of change?
I think I can you repeat the question? Sorry.
Yeah, of course. With anthropogenic pressure, what would you say is a likely pathway for sharks to evolve in the Anthropocene if they can keep up with the degree of change?
Yeah, I think a lot of these changes that are occurring could be related to climate change. But I believe that the anthropogenic pressure that we are causing by overfishing is now their main threat. Although science is studying also the climate changing aspect of it, and we are finding many detrimental effects on sharks. But I would say that the overfishing is definitely the worst pressure, the worst anthropogenic pressure, at least now.
Sure. So if we were talking about, if we think that fishing is a big pressure on sharks, do we think that do we think it's likely that sharks will evolve to accommodate any of that pressure? Or do we think that it's just...
So that's actually a thing of fisheries induced evolution, that it results in natural selection pressure to have babies earlier or more babies or things like that. They're often not as healthy. And is it possible that something like that could happen? Yes, but it doesn't. Evolutionary timescales are a little slow, and I don't think that would happen in time to be of a whole lot of use for some of these species facing overfishing pressure. But fisheries-induced evolution is definitely a thing, and I'm sure we're going to start to be seeing more and more climate-induced similar natural selection pressures.
Yeah, when we look at the fossil record, right? So sharks and their relatives have been around for over 400 million years. They've weathered 4 other mass extinctions. moderately well, right? They're still around, although not the same species and not even the same families through most of them, but they've survived, right? Even the big one at the end of the Permian at 250 million years ago, which, you know, decimated like 95% of marine life. Chondrichthyans made it through. When we look at these big trends in organisms and how, you know, the lineages that make it and the lineages that don't, organisms that are more generalists and less specialized tend to do better, right? Because if you're eating only one thing and the thing you're eating goes extinct, you're kind of screwed, right? So if they, you know, if you can generalize, or you're the type of organism can generalize either in the types of temperatures and acidities that you can tolerate, or type of food you're eating, and that sort of thing, you're more likely to make it through those mass extinctions. And so when we look at modern sharks, for example, right, just from a climate change, and the, you know, we've never had a mass extinction that's been human driven until now, so that part we don't have a record of, but we know from all these other climate change events, volcanism events, and anoxic events where the oxygen levels have plummeted and so forth, and acidity's changed, that those organisms that have a wider tolerance for all these things, they make it through. And there's no reason to think that the sharks that are like that now, at least from the climate change kind of perspective, would be okay. But again, that's not considering all the fishing stuff, right? And that's kind of that's the extra new thing that we just don't have a record for, where we can't predict other than what we're seeing right now.
Right. All right. That kind of leads into like sort of our last pair of questions here. So the first one, I'm going to give you both at the same time, because I think just the first one is too depressing. The first one really, you know, is right there. It's what is kind of the biggest threat to sharks currently? Sharks and all elasmobranchs. I do want to be inclusive of all of our rays and guitarfish and all sorts of other cool things out there. What do we think is the biggest threat? And what do we think is the best thing that people can do to contribute to conservation and make the world a better place for elasmobranchs?
So there's no doubt about what the biggest threat is. It's overfishing. It's not close. The second biggest threat is habitat destruction, and it affects 30% of the species that overfishing does. That does not, unsustainable overfishing is the big problem. That does not mean that sustainable fishing can't exist or doesn't exist, because it can and does. But what people can do, the biggest thing that people can do to help sharks and help the ocean, is don't eat unsustainable seafood. That doesn't mean, I'm not saying that you need to give up all seafood entirely and become a vegan. You can if you want, but there absolutely are environmentally friendly, sustainable seafood options that exist. Other things you can do that will help the ocean, but will not necessarily help sharks. Use less single-use plastic, reduce your carbon footprint, all of that good stuff. Donating time and money to reputable nonprofits. Shark Advocates International is my personal go-to. So there are lots of ways that people can help. And several times a year, though not currently at this exact moment, there are opportunities, especially here in the United States, for US citizens to submit public comments for or against policy proposals through our National Marine Fisheries Service, NOAA Fisheries. And if you follow on social media, Shark Advocates and or me at Why Sharks Matter, we always share those opportunities for people to get involved. But I give a lot of public talks, and people always ask me, what can I do right now? And I say, let's see, it's September. In February, there's going to be something coming up. And people hate that answer. So follow, and we will keep you posted about ways you can help.
Great. Anybody else want to add anything? Give me a wave.
I think another important thing, just like Dave Shipman said, is like the destruction of the habitat. Here in Brazil, we have the construction of several dams for the hydropower, and it has a huge impact on the fauna of fishes, because people think, they are inside the water, they're not going to die when you build a dam. But we have changes in pH productivity. We are talking about different kinds of physics of the water, if you're considering rapids or not. So we are facing the extinction of several species of fishes in Brazil, including freshwater stingrays. That is a really important thing that we have to start to consider. I think the most important case or recent case that we have in Brazil is the construction of the dam in the Xingu River. So if you want to know anything more about it, you can read in biologists in Brazil. They are with a big fight in their hands. Yeah.
All right. Anybody else want to add, or you feel good about that one? All right. Well, I think we're coming on to the end of our hour here. So I wanted to give some time for people to, take a couple of seconds to talk about where we can find you for more information about your work, or, the stuff that you're working on, or the things you think are important. Go ahead and, take a couple of seconds to plug yourself.
I can go. So I am on Twitter at Whitenack Lab. I'm not on Twitter every day and all that because I teach a lot, and so I do a lot of that. And a kid who's come in half a dozen times, I think, during this time. I kind of come and go in that. But you can also Google my name, so Lisa Whitenack, and especially recently, David and I have been collaborating on a couple of things, but one of our big things hit recently was our work analyzing the last 30 years of Shark Week episodes. So it's been all over the internet lately, and so you'll probably come up with a couple of those net papers in review right now. So yeah.
I'm at Why Sharks Matter, all one word, on Twitter, Facebook, and Instagram. Why Sharks Matter is also the title of my upcoming book through Johns Hopkins University Press. It'll be out next spring. And I'm always happy to answer. If I didn't get to your question today, or if you have a question later, I'm always happy to answer any questions folks have about sharks.
I have a page on Instagram with my students that is called Sharks on the Beach, Tubarões na Praia in Portuguese. And we also have another page there that is called Fishes UFBB. It's from the institution that I'm right now. So you can see any kind of publications that are coming, or anything that we're doing on our day by day on the lab. So you're more than welcome to follow us.
Yeah, I also have an Insta page with my new project, which is shark.rescue on Instagram, so you can follow that. And on Twitter, you can find me on Mary__right. And I've also added my email address in my talk. If you want to, if you have any questions, feel free to contact me.
Great. Okay. Well, I think we are at 3:59. And I think we've answered some really great questions today. Thank you again to the Elasmo Week organizers, and thanks so much to all of you for being here. It's been really great talking with you.